Hi, everybody. Let's properly do the angiosperm life cycle. We went over a little bit of this after your quiz the other day, but I don't want to shortchange the angiosperm since they're so important. So these are all the flowering plants that you know about. Let's see. All right, so the angiosperms are the ones that have the flowers and they produce seeds that are inside of the carpels. This is the largest and the absolutely most diverse phylum of the plant kingdom. We think there are over 280,000 different angiosperms. So when we say that it's the largest and diverse, it is a huge group. So this is in the genus, or the phylum, sorry, Magnophytata. And so that's really named after the magnolias. We think they're a primitive type of flower that were probably around when the first angiosperms emerged. They are heterosporous, and so in the case of the female gametophyte, they are just confined to being a few cells. They're completely inside the sporophyte. They're completely dependent on the sporophyte tissue as well. The male gametophyte is really just a germinated pollen grain, and there are 3 nuclei associated with that male gametophyte. So again, you can kind of see how small they've come from having, a big structure like a large fern plant, having they're diploid, but their gametophyte is an individual prothallus that you can actually see with your eyes. And now we've got these plants that are basically all sporophyte tissue except for these few cells. So let's talk about the development of the megagametophyte and the female part of the structures. And so there is a megasporocyte cell. That's the same thing as a spore mother cell, and it differentiates from all the other cells in the ovule. Nobody really knows, which cell becomes that. It's random, and one of those cells then will undergo meiosis, and it produces 4 haploid megaspores. 3 of those are going to degenerate, and then one of those will go on through the process to become the gametophyte. The integumate, at this time, the integuments will differentiate, and they eventually become the seed coat, just like when we had the gymnosperms. There is a pore or an opening at one end called the micropyle, which is incredibly important because actually it's important for the sperm to be able to get inside at some point. So that's why there's a micropyle. And so there are 2 central cell nuclei that are around, and those can either become a binucleate cell where there's two nuclei inside of one, or they could actually fuse together, forming a single diploid nucleus. So sometimes they do it one way and sometimes they do it another. One of those cells will function as the egg. So the female gametophyte really is basically this large sac. Lots of times it's called an embryo sac, and it contains 8 nuclei and 7 cells. And so that's what we've got going on for the female. We kind of went through this in class the other day. And so the last figure there, 8, would be the mature female gametophyte. And so again, those nuclei were all produced by mitosis. The 3 cells at one end are called the antipodals, and the 3 cells at the other end, one of those is going to be the egg, and the other 2 will be called sister synergids. The 2 in the center are called the central cell nuclei, and that's what the female gametophyte looks like. So this is maybe a better drawing of that final process that happens. And again, the antipodals are just those 3 nuclei that are at the other end, away from the micropyle. The micropyle can be at any end. It doesn't have to be at the bottom. It could be at the top. The synergids are just the two nuclei that are next to the egg. Again, which one of these becomes the egg is completely random. It will always be one of the ones closest to the micropyle. That's because that's where the sperm has to, the pollen tube is going to grow in and disperse the sperm. So it would be near the micropyle, but it could have been either one of those synergids. Eventually, all the antipodals are going to disintegrate. The two synergids are going to disintegrate. The egg is going to be fertilized by a sperm, and that will become the zygote and then go on to become the embryo. The polar nuclei in the middle here, which are diploid, those will actually be fused with or fertilized by the other sperm. That's why we say that this is double fertilization. And then that will become triploid endosperm. So that's how you get a triploid cell. And so again, that will be there and will be able to be then the nutrition for the embryo as it grows up. So what you're seeing here is really the beginning of a seed, right? So the green tissue on the outside would be the integuments. And I'm sorry if you hear a bunch of ruckusing. My two dogs have decided all of a sudden that this is a good time to have a wrestling match. So let me pause for a minute and get them stopped. Hold on, everybody. Okay, I'm not sure how long that will last, but we'll see if they'll quiet down for a little while while we get through this anyway. All right. So all of a sudden my slides won't go forward. So if we had a lab for you to look at this is what you would have been focusing on, mostly your whole lab. You would have been looking at something like this. And so this is sort of showing that actual embryo sac there. And this is the mature megasporocyte. And so we've got the antipodals there, we've got the egg, we've got the synergids. It doesn't really show the two central cell nuclei right now. They're there, but they haven't fused together yet. The micropyle is that little opening at the bottom there. And so I think you all understand pretty much about how the anthers produce the pollen. And so pollen is a mature pollen that's germinated is the male gametophyte. And so we've got these four different areas inside the anthers kind of divided into quadrants. And those differentiate from all of the rest of the cells. There's going to be a diploid microsporocyte cell there, and that's going to undergo meiosis and produce a quartet of microspores. We say quartet because they tend to cling together and stick together. And so here's a cross section of a lily, and so there's the four different sections. And since there's pollen grains already there, you know that meiosis has already occurred. So those pollen grains are haploid as well. And so then after meiosis, the things are going to change quite a bit, and those microspores are going to develop, and some of these happen within the anthers, and some of them happen after the pollen has left the anther. So the microspore is going to divide by mitosis. And then after that division has happened, then all of the microspores kind of split apart from each other. And so they're all individual pollen grains now. They're not a quartet anymore. A two-layered wall develops around each of those microspores. Again, we're trying to make some pollen here. And so it's going to, the exine is going to develop. And at that point in time, those microspores are considered to be pollen grains. The outer wall is the exine, and that's a protein coat. Again, it's there primarily to help with the plant being able to identify which type of pollen it is. So when this pollen ends up on a stigma, however that might happen, the stigma can recognize whether it's its own pollen, whether it's a different species of pollen. And so that is all done by that outer wall. And here's a really good picture of that extra wall. So it's sometimes they're very, very unique looking, can be different colors. You can see that this particular one has 3 aperture openings where those seams are. And so that is where the pollen tube will push its way through once this pollen grain germinates. And so here's just an array of different kinds of pollen grains. So you understand they're not all circles. Some of them can be, really, really special. And like, we've got the ragweed up there at the top that has the spikes on it. And I kind of like the one in the middle. I think that's really interesting looking the way that wall is. And so these can be used, the exine can be used to help identify pollen. Pollen is kind of like having fingerprints, and it's pretty unique. I'm not sure that there are 280,000 different kinds of pollen grains, but I know we could narrow it down to at least the genus of plants. If we were looking at the pollen, we would know whether it's pollen from an oak or a rose or something like that. So we've talked a lot in this class already about pollination and fertilization. Just a reminder, pollination is just the pollen getting to the stigma from an anther, and that could happen by wind or animals or all the other things that you watched in the video. Fertilization happens later on, and that's the actual fusion, the fertilizing of the egg with the sperm. And because this is an angiosperm we're talking about here, there's actually double fertilization. So one sperm fertilizes the egg, and the other sperm fertilizes or fuses with the endosperm, fuses with those central cell nuclei to make endosperm. The length of time from the first event to the second depends on the size of the flower generally. It can be just a few days if it's a very tiny little flower, or it could be several weeks or even months if it's a very large flower. So that was one of the criticisms I had when I watched the David Attenborough video, is that he would say, the flower's been fertilized when a pollinator visited. And that's not true at all, right? It was just pollinated. It wasn't fertilized. So after pollination then happens, that's when the pollen grain will sort of differentiate a little bit more, whether it is the same species or isn't the same species. If it's landed on a young, immature stigma, nothing happens. If it lands on an old stigma, nothing happens. If it lands on the correct stigma of the correct species, at that point in time, the pollen is going to germinate and a pollen tube will grow. And so there are a lot of caveats to this pollen floating all around there. It's not a given that anything's going to happen to it. The pollen tube is going to grow down through that style and then eventually find the ovary. The pollen tube works its way into that micropyle, and then it delivers its sperm right next to the 2 synergids in the egg. And so that's what's happening with the sperm. So there's a quick video there, and again, this is on your website on Canvas under files if you want to watch it, but it basically just shows a pollen tube coming out, and you guys, most of you got to see that when we looked at it in the lab a while ago. So there's again the pollen grain. This is the mature male gametophyte. Different shapes and numbers of apertures depending on what kind of pollen it is. Those 2 sperm cells there have divided from one spermatogenous cell. The pollen tube's growth is being controlled by that vegetative cell, and you can see where the vegetative nuclei is down there. These pollen tubes are some of the fastest growing cells that we know of. They really grow really, really quickly. So once the pollen tube reaches the micropyle, it continues to find the female gametophyte, and then its 2 sperm will be released, and boom, we have double fertilization. This is the only group of plants on the earth that does this. And it's pretty remarkable because this is a completely different way of making nutrition for the embryo, right? We have endosperm. I think that's just what that says. So let's look a little bit closer at this. So there are the integuments that will eventually become the seed coat. The endosperm here is triploid. You should know where those nuclei came from. The zygote here is diploid because it came from the fusion of the egg and the sperm. Those antipodals that are at the other end and the two synergids at the bottom, again, they just disintegrate and they don't become anything. Eventually, if somebody hands you a seed or you're looking at a seed, this is what you have. You have the seed coat, the embryo in the center, and all surrounded by endosperm. And so I think that's kind of interesting how all these different pieces and parts come to be. So if we look back through fossils, we know that flowering plants occurred about 160 million years ago, right during the late Jurassic period. And primitive flowers were thought to be sort of bowl-shaped and have just kind of flowers that were petals that were kind of spirally arranged. They weren't fused together at all. They didn't have any particular pollinators or anything like that. Those features took a long time to evolve, but they were just very, very simple kinds of flowers. They tended to have both male and stamens and pistils, male and female parts. And again, those are all considered to be kind of primitive features. All of the weird, bizarre things that you saw for like orchids, those came along much later. There's a very primitive flower, what we think kind of looked like, and again, nothing's fused. There are no landing platforms or any of the other cool things that you got to see in the video. So let's see if we can get this to work. Let me click on this YouTube video real quick. Actually, I'm not going to bother doing that because I'm going to post this on YouTube.
This unforgettable vacation memory. It didn't actually begin.
I'm going to post this on YouTube, and YouTube doesn't let you link to other YouTube videos within a video. So why don't you watch this from your file, and you can kind of see how flowers actually have developed over time. So again, I mentioned earlier that there's over 280,000 different ones, and really, why do we have such huge diversity in the flowering plant kingdom compared to all of the others? There's just so much more diversity here. One of the answers can be seen right here in this picture, right? So we really are amazed over time about how the flowering plants have developed all these amazing different pollinators, relationships with not only pollinators, but relationships with different kinds of dispersal agents. And then, their seeds are everywhere. They have all those special adaptations for keeping their seeds dormant when they need to be dormant and germinating at the right time. And so it's not a wonder that there are that many different kinds of flowers when you think about all of the different relationships. So there's a quick overview of the life cycle. Please refer to that handout that I gave you in class the other day that has the life cycle kind of drawn out for you too. And again, this info will be on your written exam next week. Okay, I'll talk to you later. Bye.